Welcome students. Welcome to my channel, that is BioMinds. Today the lecture is about antibody. We will discuss antibody in different parts. Today lessons we will discuss the structure, history and the function of the antibody. So first we start with the introductory part. What is antibody basically? Before that we should know what is B cell. Okay, so B cell which secrete your or which produce your antibody. Okay, so B cell matures in the bone marrow. Okay, and that produces antibody. So there are basically two types of antibodies: membrane bound antibodies and secreted antibodies. Okay, membrane bound means what? Here in this diagram you can see. So this is the B cell. So this Y-shaped structure is the antibody which is attached to the surface of the cell surface of the B cell. So that is known as a membrane bound, means it is attached to the membrane of the B cell, and that is known as the B cell receptor. Okay, and with the antibodies which are being secreted in the circulation, they are known as a secreted antibodies. So B cells has two types of antibodies: membrane bound antibodies and secreted antibodies. So whenever there is an bacterial antigens or any antigens or any foreign particles enters your body, it will go and bind to the this BCR, that is the B cell receptor. In short, we can call it as a BCR. Okay, that is your membrane bound antibodies, and it will activate your B cell. So once B cell is activated, it will differentiate into plasma cells or memory cells. So plasma cells that secrete the antibodies in the circulation, that is in the blood circulation, and memory cells will maintain the memory of the infection. So this is how the B cells produces antibody. Okay, so the B cells and the antibody mediated immunity is known as a humoral immunity. Okay, because it is present in the circulation. Okay, in the blood circulation. So next topic comes, how does this B cells, how does this antibodies is being discovered, or how scientists got to know that it is present in the serum. Okay, serum of the patient. So these two scientists done the experiment. The scientist names are A. Tesselus and E. A. But in 1939, they have done very classic experiment for identifying that in which fraction of the serum contains the antibodies, or they have discovered that there is something in the serum of the patient which can fight with the disease. Okay, so what they have done is, so they have taken the rabbit, they have immunized the rabbits. Immunize means what? They means some, if you inject something in some animal or in the human also, and which is foreign to that particular person or animal, then that person or animal produces immunity against it. Therefore it is known as a immunization. So vaccine, which we say, it is also immunization process, because whenever you inject something into the some person, that person will produce the immune response against it. Therefore it is known as a immunization. Okay, so what they have done, they have immunized rabbits with the protein ovalbumin. So ovalbumin is the albumin of egg whites which is foreign to the rabbit. So hence rabbit eventually produce the immune response for the ovalbumin. Okay, after that, after some few hours, okay, they have extracted a serum from the rabbit, and they have divided into this serum into the two aliquots, and they, from the one aliquot they have done the electrophoresis. So I hope you all know what is electrophoresis is. So electrophoresis is the technique where the molecules is being separated by the size, charge and shape of the molecule. Okay, the electrophoresis is a technique where the molecules of your choice is being separated by charge, size and the complexity, you can say, or shape. Okay, so they have done the electrophoresis of the serum. So after that they got the peaks. Okay, peaks. So in that, the first, which is very dark peak, which is of your albumin, because it's the albumin of egg white. So albumin peak is very high. Next we have alpha 1, alpha 2, beta peak and gamma peak. So when they have studied this gamma peak, they got to know that there is something in the gamma peak which fight with the disease. Okay, that can fight with the any disease, that has ability to fight with the disease, and therefore they are term it as a immunoglobulins. So they have found this globulins, so they have termed this gamma globulin as a immunoglobulin because it has some immune response ability or property. Okay, so you can observe that immunoglobulin is further named as antibodies. Okay, so whenever you do that experiment, an experiment using serum sample, and if you want antibodies of from that person, you can use the gamma fraction from that particular person's serum sample. Okay, so in the gamma fraction we have antibodies. So this is how these two scientists have demonstrated. Okay, so next topic is the structure. So as we all know, we have studied this. As we all know, the structure is Y-shaped. Okay, this is Y-shaped structure. Okay, so this basically the antibodies are the protein in nature. If it is protein, then it is made up of gene. बराबर. So it's a product of gene which is protein, or you can say peptide. So it's a protein in nature. Antibodies is protein in nature, just remember this. Okay, so there are basically four polypeptide chain. Four polypeptide means different peptide chain. So there are total four polypeptide chains. उसमें से we have two heavy chains and two light chains. So this is heavy chain and this is also heavy chain, two heavy chain. This is one light chain, this is another light chain. Why this is called as a heavy chain and why this is called as a light chain? As you can see, the heavy chain is longer, light chain is shorter, as well as the molecular weight of the heavy chain is higher than the molecular weight of light chain. So molecular weight of heavy chain is 50,000, whereas molecular weight of light chain is 25,000. Okay, therefore it is known as a heavy chain and light chain. Can you see the SS in this structure? So what is this SS? So SS is the disulfide bond. Okay, so your light chain and the heavy chain is joined together by disulfide bond. This is covalent bond, okay, which is stronger. Okay, so we have a dimer means two, dimer of heavy chain. Okay, one dimer of heavy chain and light chain, another dimer of heavy chain and light chain. So we have two dimers, this is one and this is another. So therefore it is the also known as the antibody is also known as a dimer of dimers because we have two dimers. Okay, एक dimer में we have heavy chain and light chain, and another diamond also we have heavy chain and the light chain. So these are made up of two dimers, therefore it is known as a dimers of dimer. Okay, so in this antibody structure, can you see this VH, CH? Okay, what is this VH and CH? So V means variable region, C means constant region. So variable region means what? With the this is protein in nature, so protein is obviously made up of amino acids. So in the variable region of the antibody, the amino acid sequence changed. Okay, it will change according to the antigen. Okay, because it has capacity of antigen binding. So variable region of heavy chain and variable region of light chain, amino acid sequences of both of these regions changed. But whereas the constant region of heavy chain, this is constant region, the light purple color and of heavy chain, and the red one is constant region of light chain, which does not change, which will remain in same in all the antibodies. Okay, so therefore it is known as a constant region, and this above four regions are known as the variable region. So it has antigen binding capacity. Okay, we will see this in the upcoming classes. What does this heavy chain and the light chain? Okay, so this is the overall basic structure of the antibody. Okay, so how do scientists get to know that this is the structure of antibody? So because they have done some experiment to confirm the Y-shaped structure of the antibody, or to confirm there are two chains. There are total four chains. Okay, so they have done certain chemical and enzymatic methods to reveal the basic antibody structure. Okay, so they have. Okay, so peptidization, we generally get three fragments, two identical and one different. So for the identical fragments, the molecular weight of identical fragments are 45,000 each. Means there are two now. So one is 45, another is 45. One fragment which is generated, which is not identical, with the molecular weight is 50. So the two identical fragment has antigen binding activity. So it has something in the structure that can bind to the antigen. Therefore, it is known as a Fab fragments. So just remember this Fab fragments. Whenever you study antibody in further immunology class, you come across these terms very often. Fab, Fc fragment, Fab fragment. Okay. So Fab fragment which has antigen binding activity. Therefore, fragment of antigen binding. Fab. F stand for fragment, A antigen, B binding. Okay, so the two identical fragments are fragment of antigen binding. Next, we have one fragment which does not have any antigen binding activity and that can be easily crystallizable. Therefore, it is known as a Fc fragment. Fragment, F stand for fragment, C stand for crystallizable. So, peptidization gives you three fragments, two identical and one different. Next, we have pepsin digestion. Pepsin digestion gives single fragment of 1000 molecular weight. Okay, so they have two Fab-like fragments designated as Fab2 fragment, and Fc fragment was not recovered because it is digested into multiple fragments. So in the pepsin digestion, you can able to get only Fab fragments together in the one, that is 1 lakh molecular weight. Okay, mercaptoethanol reduction gives, it cleaves the disulfide bonds. Okay, whereas pepsin and pepin cleaves, it is a protease, so it gives the peptide bond. Okay, mercaptoethanol, whereas it cleaves the disulfide bond. I have told you disulfide bond is there in your heavy and light chain. Okay, therefore there is a separation of heavy and light chain when you do the mercaptoethanol reduction. Here you can see in this diagram. So this is the basic structure of your antibody. Here we have Fc fragment. Here we have, so after the peptide digestion you get two fragments. Okay, one is Fc, or three fragments, two Fab fragments. Mercaptoethanol, the disulfide bond, this disulfide bond is being cleaved and you get four chains, two heavy and two light chains. In pepsin digestion, you get this entire Fab fragment together because it will cleave in here, and Fc fragments which is crystallized or uh, separated into the multiple fragments, so it is not being recovered. Okay, so this is all about the today's class. Okay, so here we have discussed structure, history, and the uh, structure, history, and the chemical and enzymatic treatment. Okay, so I have created one test for you. So I have posted a link of the test in the description box. So I encourage everyone, please give the test so that you can understand. I got to know how much you have understood the topic, and you will also get to know that you have understood the topic or not. Okay, thank you.